So thanks for coming out. I'm super excited to host today Smita and David, who are visiting from Columbia. Yale, sorry. Oh my goodness. I was jumping ahead. Smita did her postdoc at Columbia with Don up here. So she's been at Yale for the last 3 years now. I was at Columbia before that. So I'm off to a good start. Maybe I should have stopped now. But no, so they're going to talk a lot about high-dimensional learning for single cell in particular. And as I said, Smita came from Donna's group at Yale before that.
In Columbia.
I'm getting backward for all these ones. I really should stop now. Before that was the University of Michigan, or maybe it was University of Oregon or something. I don't know. David was at University of Amsterdam for his PhD, but he actually spent most of his time at Weizmann, which is right next door in Israel, working with Eran Segal, and is now a postdoc with Smita at either Columbia or Yale or one of those two. So anyway, I'll stop and let you guys. There's folks on Skype too as well. So
okay.
For those of you that are on Skype, I will get on. If you want to ask a question, type it in and I'll relay it.
Okay.
Thank you.
All right. I'm gonna go ahead and get started. If you have any questions or if you want to know more about something, I guess either you could ask me right away or you can ask me at the end, and then David has extra slides on everything if you want to go into more detail about anything specific. Now I'm just going to talk about basically what our group does. We're jointly located in computer science and the medical school at Yale, and we're basically a machine learning or applied math type group, and we develop techniques that are meant to analyze complex biomedical data and can usually or often be applied to other types of data, because we believe that the bar for analyzing biomedical data is a little bit higher because there can be all kinds of sparsity, missing information, very, very noisy, a lot of nonlinear effects and things like that. So we believe that if basically we do unsupervised machine learning techniques, so if we're able to detect those sorts of patterns, then hopefully we can detect other sorts of patterns as well. So, as I was mentioning, our group develops techniques that deal with data that can be very high dimensional, so you're measuring a lot of different features. Often we apply our techniques to single-cell RNA sequencing data where the features are transcript counts, mRNA transcript counts, but we've applied it on a variety of other data sets. Also high throughput, so there's a lot of different units, so we've tended to work with single-cell data where the units are cells, or data where the units are patients, but there's a lot of them. And there's a lot of heterogeneity within the data set, which means that we want to learn what the different ways that the data itself is stratified, what the different patterns are, how much information and redundancy there is in the features. The challenges this kind of data can pose is, as I mentioned, there could be sparsity, even though there's many, many features. A lot of them could be missing for a lot of the data points. There's a lot of noise, so there could be very extrinsic noise, background ambient noise, or there could be specific types of noise. There's nonlinearity in sort of the shape of the data and how the features relate to each other, and just the scale of it can pose computational problems, so the algorithms need to be scalable. So with this kind of data, one question that we often ask is, how do we look at this data? So how you might have a lot of data, and often you might not even have any ideas what your data can tell you or what can it even predict. So in this circumstance, we have unsupervised techniques that will help you just sort of look at and explore the data to even generate hypotheses from it. Some of the technologies in biology that are generating this scale of data is mass cytometry, or CyTOF, as you mentioned, which generates a lot of protein channels, RNA sequencing, high-C TCR sequencing, which I know a lot of you work with, and sort of the list goes on. We think this kind of thing just keeps exploding, and there's more and more technologies that are capable of generating this. So how do we sort of deal with these challenges? And one of the themes that we've sort of seized upon in our lab is the fact that even though the data is measured in a high-dimensional space, it has intrinsically low-dimensional structure. So, for example, in the gene single-cell RNA sequencing world, there are a lot of genes that you measure, but these genes have a high degree of informational redundancy between them. And as a result, the cell types and states are constrained. So there's an intrinsically lower dimensional shape here. So the ambient space was a lot bigger than the intrinsic shape. And so that's one of the principles we use in our lab. How do we learn this lower dimensional intrinsic shape? As you can see, this shape can be nonlinear. How do we understand this shape? And one of the fields in math that we use a lot is graph signal processing, where there's a nice theory of manifolds, and we show that we can also implement the same concept with deep learning, where not only does the data have shape, but there is some smoothness in it. So the smoothness or the locality in the data can really help you learn and understand this shape. I'll give you sort of an example here. Smoothness meaning your data points are occupying the state space in some semi-regular fashion, even if they're not uniformly sampled across it. You have samples in different parts of it so that you can learn something about it. It doesn't have to be one smooth shape, it can be a collection of them. So basically, if you have this kind of highly nonlinear, lower dimensional structure, how do you learn it? The very basic way you learn it is you look for very similar data points to every data point. And to some level of granularity, even if you have high dimensions and noise, you should be able to tell data points that are very similar to your point in a very local way. So if you're trying to look for points that are very far away, you can get lost, because unless you know this is structured like a Swiss roll, you might not understand that the points in the different arms are actually far away. But you can understand which points are very close to each other. So once you have points that are very close to each other, you can basically take a walk by taking small steps to local neighbors. And this walk will somehow help you clarify the shape of the data and find this highly nonlinear intrinsic dimension that the data follows. So when you walk in small steps, you can define a Markov matrix that says, starting from any point, you're going to transition to other points based on your measure of similarity. That measure of similarity doesn't have to be necessarily very perfect. It can be pretty rough, but as you walk longer distances, the probabilities add up so that the longer you walk, the more you hone in on the main dimensions of the shape. So this concept is called data diffusion, or a random walk, and this data diffusion type concept where you find these main nonlinear eigendimensions turns out to be the sort of basis of graph signal processing. So we used this concept for the first project that David and I did together that was just published in Cell, and it's an algorithm called Magic. And what Magic shows is that by learning this manifold of the data, you can restore values to cells that were missing, or you can denoise cells such that you can further process them. So restoring data basically to the main manifold dimensions. The motivating problem was in single-cell RNA sequencing, which came out about 3 years ago. You have a cell-by-gene count matrix, but most of it's missing just by the way RNA capture works. It's like reaching into your cell and grabbing 5 to 10% of the transcripts, and the rest of them you leave in the cell. So given this, a lot of what I'd done with CyTOF, for example, in my postdoc, which is try to learn relationships and signaling the relationships between proteins, could no longer be done. So if you're looking per gene now, and you're trying to get information from your 20,000 genes as far as which gene has a relationship or correlation with another gene, it becomes very hard to discern this because you're not seeing both the genes in the same cell at all. So if you only see one gene in 10% of the cells and another gene in another 10% of the cells, the likelihood of you're seeing both genes in the same cell is 1 in 100, and so a lot of this data is dropped out, and you can't do the type of gene regulatory analysis that seems to be promised by the technology. So the steps of magic basically are that you start with a cell by gene matrix, you calculate distances, but again, you don't trust far away distances, so you localize the distances by passing it through a Gaussian affinity. So that gives you very local neighbors, and then you Markov normalize it to create a random walk matrix, and then you exponentiate this matrix to compute the probabilities of longer random walks. And then this has honed in on the main dimensions of the data. So if you eigendecomposed it, you'd get what's called a diffusion map, but we don't actually eigendecompose it. We basically project this operator onto the data to learn a weighted average of manifold intrinsic neighbors to get the imputed data. So you basically have data before magic, and after several steps of imputation, when you project, you get your restored data after magic. You can see some of the other types of noise in the data besides missing values are also restored. And what the subsequent steps of diffusion do are it connects the manifold more and more globally. So you get more global directions. And as you do this, you take off the low noise dimensions and you hone in on the main manifold dimensions, and you see restored relationships per gene. And it can restore it to nonlinear values because you're not imposing any sort of global constraint on the shape of the relationship or what you impute.
Question.
Yeah.
What is each black point signifying?
It's a single cell.
It's a single cell, but what's the x-axis and the
They're two genes, just two random genes. You can think of it as.
So this is single cell RNA sequencing. So you count molecules per genes in a cell. So these are molecule counts, observed molecule counts.
Yeah, I try to understand like what on the x-axis, like how do you determine the coordinate?
Oh, it's expression level.
Oh,
so it's just two features. So you can think of it. We had a matrix with a bunch of missing data, and I'm trying to learn the relationship between two columns of the matrix, just to see if the two columns have correlation. So if you had people, I'm trying to figure out if there's correlation between their, I don't know, height and salary or something like that. But I just couldn't do it because most of it was missing. And when I restored it, I can restore the relationship between these two features.
So I understand like at the high level is basically you have a sparse matrix and you try to do matrix completion, right?
That's right.
But actually it's not matrix completion, it's a harder problem.
I'll talk about why it's not the same as matrix completion. So matrix completion assumes that the values you have in your matrix are okay, and you just have to compute missing values. But that's not this situation. This situation, you undersampled everything. So you have a small set of samples from your cell, and everything is undercounted to some extent.
Even the non-zeros you can't trust.
I see.
So if I understand this correctly, you actually can't even tell what's a missing value.
Exactly.
Expressed.
Yeah, there's no distinction.
Yeah.
And the algorithm doesn't distinguish like that either.
Although if something's highly expressed, the chance that it would be zero is pretty low, right? If you've got something that's got thousands of transcripts.
Well, usually if we see a transcript, it's pretty trustworthy unless you have a mapping problem.
Yeah, no, but what I'm saying is that it's not even random in the sense that if something's got lots and lots of transcripts, and you have a 10% chance of taking each one, and if you have a 10% chance of taking 1,000 things, the odds are going to get zero or they're minuscule.
Yes.
So
yeah, if you have a transcript that dominates, or
you'll see it.
You'll see.
You know, if you have cancer cells that go berserk.
Yeah, you might see the cell cycle genes or exactly something like that. So then you're just
actually the interesting genes that are low expressed, like transcription factors.
Mm-hmm.
and surface
And
markers.
uninteresting stuff that is highly expressed.
Yeah,
unfortunately.
Yeah.
Actin is highly
Actin,
expressed.
yeah, there's a lot of actin.
Yeah. So exactly. Our cell surface markers, because then you can use them to sort and study cells. Those are exactly missing before your imputation. So did that help answer your question? Yeah. And I'm not assuming you know what these relationships look like, although if you know about epithelial-to-mesenchymal transition in breast cancer, you know that this is the correct direction of these relationships, because snail is a transcription factor and vimentin is a mesenchymal marker. So when snail goes on, vimentin goes up. And then ecodierin and vimentin are markers of epithelial versus mesenchymal state, so they're not on at the same time. So.
By the way, I don't know if you have a slide on that. We did try, matrix
I have a slide on
matrix completion. We did try matrix completion techniques, and they totally fail on this type of thing, just so you know.
We always try to find ways to validate it computationally and otherwise. So here we had a gene of worms that were developing for the first, I think it's 48 hours. And they had bulk sequencing, but there was a natural sort of manifold structure that we knew about. And we artificially dropped this data out and we re-imputed it with magic and denoised it, as you can see. And the original relationships got totally messed up. Most of it sunk down to 0. And then you re-impute it with magic and you see that the original relationship is restored or even further clarified. So why is this interesting to us? Because you can learn a lot about individual genes now. You can look at their pairwise relationships, their individual probability distributions are re-represented, and you can distinguish between different populations and how much of a particular gene is represented. And in addition to looking at individual genes, the overall data is restored. So this is before magic, and afterwards you see a very clear structure of this data. This is again data that's mimicking the epithelial-to-mesenchymal transition on breast cancer cells. So we see different structures corresponding to the yellow is mesenchymal cells that successfully transitioned, for example. So one thing we can do after magic that we completely couldn't is do something called archetypal analysis. So there's data before magic and after, and the main idea is to fit a polytope to the extreme ends of the data to figure out what's creating the overall shape. And when we did that, we were able to identify sort of extreme endpoints of this shape that correspond to chromatin modification. So for example, now biologists could look at these intermediate states that people hadn't really considered that much and see that there's a sort of long program of chromatin modification that's going on. So a lot of times people hone in on the first part of this, which are epithelial cells, or the last part, which are mesenchymal cells. They're not really sure how these cells transition through the intermediate populations, and that's something that you can start to get a picture of after you've restored all your gene values. This is comparing magic against sort of matrix completion and low rank approximation methods. So you can see that NNMC is nuclear norm-based matrix completion, the Candice-based method, and it doesn't perform super well here. Low rank approximation actually does somewhat better, but it's a linear method, so it's not completely denoising in the nonlinear directions. And then you see magic restores sort of the data shape to something that's identifiable to us. We measured it against a lot of other ways. So people often ask us, do you need this sort of complicated random walk technique? Can't you just do this by imputing with KNN? And you see it's not nearly as good if you impute with just your k-nearest neighbors. The global manifold connections actually do make a difference when you're learning the shapes. And you can't use individual diffusion components or anything like that to get the same. So it can't be sort of just simple smoothing on your graph. So the manifold learning actually makes a difference. So after we did this project, so basically what we did was we restored the gene values. We thought we could do similar inference because we've learned the gene cell graph on external variables, and we thought this could really help us because single-cell RNA sequencing data now is not only in a single condition, but it can be generated in multiple conditions, and now people want you to compare conditions. So I have the single-cell data, and I have it with perturbation. So what's the real difference? And measuring the difference actually, pretty hard because first off, there's all this noise that sort of occludes the difference. But also the measurement effect itself could be weak or could be measured weakly. And so we thought we'd actually treat this experimental variable itself as a similar signal as we treated a gene and use the same sort of graph manifold denoising technique on an external variable, but it's configured a little bit differently because we don't know some things about the experimental variable. So this is a technique that's in preparation. So David is sort of supervising this project, and Jane and Dan are working on it. The idea is to smooth an external, perhaps experimental label on the data graph to impute sort of continuity and infer associations. So the idea here is you have cells from condition 1 and condition 2, but you've learned your graph from single-cell RNA sequencing data from the whole data that on the genes. And now you can sort of interpolate the labels to get a continuous variable that can now give you some enhanced abilities to tell the difference between the two conditions. So one thing it can do is it can improve your ability to correlate variables with the experimental condition. So before you had these discrete labels, and between condition 1 and condition 2, is there really a significant difference? Can you tell? Often you can only tell if it's a monotonic relationship like in A, but you can't tell a non-monotonic relationship like you have in B. And so after we call these melding, we've turned it into a verb, so we're melding variables. After we meld the variable, we infer this latent dimension that seems much easier to track to see if it correlates with the experimental condition. So you can think of the meld dimension as a continuous experimental variable. The other thing you can do is help us identify cell types. So if you have condition A and condition B and you make samples from it, you can see which cell types are enriched in condition A versus condition B. So some of sort of the math behind it, they're also using graph signal processing. So the idea in graph signal processing is you have a graph and you're treating this variable as a signal on that graph. And what you're actually doing is you're low-pass filtering this graph. So like before, when you power the matrix, what you're actually doing is you're keeping the high eigenvectors of this Markov matrix, which range from 1 to 0 by the properties of Markov matrices. So if something is 1 to 0 and you raise it to a power, it goes down unless it's close to 1. And that's the property that you're using that's sort of smoothing on this graph or smoothing on the Laplacian. So you see that before the smoothing, the signal is kind of noisy. It has all these high-frequency components. And as you smooth more and more, you hone in on the low-frequency components. So you're smoothing the external variable along the main directions of the graph, assuming that the main directions of the graph sort of represent the difference. What? T would be how much you power the matrix, even though in this setup we actually don't power it. We have it as a convex optimization that trades smoothing with reconstruction. But in magic we had a T where you powered it.
How strong your filter.
How strong you power, how much you power, or how much you smooth. For example, if you have time series data and you're looking at the expression of SOX10, this is some embryonic stem cell data, you impute continuity to this, and then you can tell there's two cell populations, and one cell population rapidly increases the expression of SOX10, which is a marker for some lineage. I can't remember which one right now. And so in these kind of inferences that you can make. Maybe even more useful kind of inference we can make. We're testing it on this HIP-C data. So Yale's part of the Human Immunoprofiling Project Consortium. So we get basically CyTOF4, this is single-cell RNA sequencing data from these different infectious diseases. So this is Lyme disease analysis. This is the difference between a patient's first visit and a patient's second visit. So we meld the visit label, and what we're starting to see is the right side is colored by the melded time. So the populations that are enriched at the second visit are in the lighter colors. Populations that are more common in their first visit are in the purple colors. So you see that T cells, there's certain kinds of T cells that are showing up in yellow. And if you sort of zoom in on them, you see in that the second visit, there's certain regulatory T cell populations that are appearing. And regulatory T cells are actually quite rare, and it would be hard to pick this out without smoothing the coloring on the label. And they're memory T cells that are upregulated in visit 2. And we can generally see how these populations arise and then maybe go away with the different, as the patients come back, and they're expected to come back more and more time. So we're excited about using MELD for this.
So here you have like two labels. So like, if I understand correctly, so like you view it as
you can have more than 2 if you have time steps, like you can have 4 or 5.
But in this particular case, it's kind of like you view it as like 0 and 1 originally. You have discrete 0 and 1. Now you do the matrix powerings. Eventually you get a little bit. It's kind of a little bit like diffusion to some extent.
It's
like
exactly like diffusion.
Okay. So you have a soft probability in some sense.
Yeah, you have a soft probability that in some respect you could interpret as sort of a likelihood that it was generated by condition 2 versus the condition 1.
But then in specific settings, the score can be interpreted as something. For example, these are 2 visits in time. So now you can say that we imputed the time, right? Though you measure 2 time points, we effectively get many time points. But it's still a score of how likely that cell, came from
one
sample
condition.
1 or sample 2.
I see. So when you generalize to multiple condition, then it will become a multinomial.
Yeah, it would be a multidimensional impute. And you can do the same thing, right? If you encode your condition with 10 bits, you can meld all the 10 bits.
But you do have cells from sample 1 and sample 2.
Yeah.
So what you're doing is you're hallucinating what happens in between, right?
Yeah.
Or imputing what happens.
Yeah, you're imputing what could happen in
physical
between.
analysis, you haven't changed the situation at all. If the doctor wants to see whether there is an uptick in regulatory cells, they would still compute the same p-value.
So basically, that's not exactly correct. So even if you have 2, you can see if there's a strong regression with a condition after meld that you couldn't before.
And there's also some unique information.
So imagine computing p-values to see if the distribution of some gene is different. Like in this condition A versus condition B, you'll do some sort of statistical test. But I'm saying you can do some kind of stronger inference on the meld dimension. You can compute the correlation with the condition.
Moreover, A and C, you could, in theory, get the original data, right? As you can see. But B, you would not get the original data. It's up, down, here.
Can we zoom in on that for a second? So what about the data that you see in panel B would lead you to conclude that?
So imagine, so you have to understand that each sample is very heterogeneous, right? So there are some. So let's maybe go in reverse. Let's assume that this is the original. This is the ground truth, okay? This is the original system. But now you measure that in 2 samples, right? So you cut it into 1 sample and 1 sample here. The result will be this measurement, right? But there will be some cells here and some cells here that were actually here at the peak. But you're missing that information now. But with MELT you can recover that.
And you're using similarly in other genes as well to figure out that which ones are closer to 1 and 2 versus farther away, because other genes are changing also.
Yes.
Exactly. You learn the manifold on the gene expression, right? And now you use that manifold to impute, to interpolate this external
data. You're relying in some sense on the noisiness
So
of
then
the
how do you compute the p-value?
Yes.
How do you compute the p-value then? How do you say?
We're not super focused on p-values. We don't believe in
No, I
them.
know, but
In single cell data, it's actually been shown you can have any p-value show anything because everything is rare. Because you have thousands of cells and 20,000 genes, so anything is significant.
How do you do this? Is it a visualization tool where they look at this and say, well, maybe this is happening, so we should have more samples in between?
Yeah, you could, or you could
Or do
see
they
if
say,
you think.
this is enough for me to be convinced that this is what's going on, rather than really?
I don't know. It's up to the
You could do
clinician.
statistical analysis on top of this if you wanted. It depends on
Have
what
you
your
tried?
your
I mean,
goals.
how would you do it?
No.
No.
Well, so one thing you could do is, so you originally measure in 2 conditions, you do meld, and now you gate this into 3 bins, and now you have 3 conditions, and now you can do the same things that you could.
So that we've done.
originally do,
We've
but-
validated it by doing that. We've left out a time point that was actually measured, and we've melded on the 2 time points that are flanking it, and we can impute back the middle one.
Right.
So we've done validation.
Right.
But we're not super big on statistics because we
Well,
realize
but,
we can lie
you
with
can-
statistics.
You can,
Doctors are, so.
sure, but you
Well,
could,
maybe they shouldn't be.
you could generate some kind of null distribution where you randomize things, and then you could get a p-value.
But it's
There are
very
ways
easy over easy to get good p-values that way. So I will highly
Yeah,
warn you about that.
randomization will give you the p-value.
Yeah. So if you will do meld on a random manifold, for example, so like a random graph. Yeah.
So another project that we did that sort of uses these same ideas is we realized that we have all of this high dimensional structure, but we don't have a very good way of visualizing this structure for exploratory purposes. So if we, in some cases, we knew genes we were interested in, so we were visualizing biaxial plots. In another case, we visualized PCA But it came to our attention that all of these methods sort of leave out a lot from the data. So because we learned the whole manifold of the data, there should be some way of visualizing it that preserves structure a bit more. And this led to a project called PHATE that's under revision now. So the idea of PHATE is that because we have a natural understanding of the shape of the data, we can squeeze it into lower dimensions in such a way that captures both local and global structure, and therefore sort of trajectory structures in biology, but also disparate cluster structures. And we realized that not a lot of other visualization methods really preserve structure in this way. So I'm going to show this both on artificial data and data generated from an embryoid body differentiation system, where you start out with human embryonic stem cells, and then you differentiate them into a variety of different lineages over a 30-day time course. So this artificial tree data, which my lab sometimes calls the dumb tree, but you'll find out that the dumb tree is harder than it looks, as you see here. So if you take this dumb tree and you add noise to it and you rotate it in high dimensions and you look to see if you can visualize it, if you try to visualize it with PCA, you're missing a lot of the structure in the dumb tree, even though it picked up sort of the global directionality in some way. It is going from blue to green, but it's losing a lot of the branching structure. tSNE seems to be all the rage in this field. I'm not sure, maybe in other fields too. But tSNE actually sort of shatters the data in a bad way because it only cares about keeping stochastic neighbors together. And we believe that if you don't keep just neighbors, but keep manifold intrinsic relationships all together, it's a better way of preserving the structure in the data. And you see, PHATE can preserve the data, but it can also denoise it, which is important in, so you see the lines that PHATE gives are thinner. You can also denoise it in the manifold intrinsic dimensions. So here's PHATE shown on, and we especially hope that a lot of people start using this because this basically can work on any type of data. Here you see the embryonic stem cell differentiation system. The red to the left is all embryonic stem cells because that's from day 0 to 3, and we started with embryonic stem cells. And as the days go on, you create the lineages that are colored in orange that we picked up on days 6 through 9, and then finally the blue were the latest to be generated, 24 through 27. And again, you see PCA kind of picks up at least this time label, and the fact that there's fewer cells, so there's less space occupied by the red than the green and blue because these are branching out as lineages. But the internal structure is not clarified. But tSNE doesn't even do that, so this is very bad for preserving continuum structure. So you see the red is shattered out to on either sides of the green, and the global relationships are all completely messed up. So you cannot trust this for understanding really the overall global structure of your data. Diffusion maps, which is somewhat based on this concept, but it's an older method. It doesn't preserve in a way that you can visualize. Basically what it's doing is it's taking each branch, each trajectory, each cluster, and putting it out to a different dimension. So you need to look at many diffusion dimensions to get at this, even though it has some of this information. And this is how the PHATE algorithm works. So the first 3 steps are similar to the steps that you saw before, which is you have the data, you learn distances and affinities, and you power the matrix, although usually to a higher power because you're visualizing in 2 or 3 dimensions. But then we use some information theoretic techniques after that. So after that, we realized that we want to really preserve the information in this matrix, sort of information theoretically as much as possible in low dimensions. So we come up with an information theoretic distance between these diffusion probabilities. And then again, we don't want to lose any information. So instead of just visualizing eigenvector 1 versus eigenvector 2, which is what people do in PCA, we squeeze all the variability into 2 dimensions by running metric MDS on these informational distances that we came up with. So we're making sure that we don't leave any information out there that we haven't captured, and that we're capturing as much as possible in our distances. And the idea of using an information theoretic distance is that each point is a probability distribution. each point is a distribution between affinities, or probabilities of walking. And if you just took a straight pointwise distance, it would be dominated by what's happening with the nearest neighbors, and it would look a lot more like tSNE. But if you use an information theoretic distance, usually information theoretic distances have a damping factor. So you have a log, as you do in Shannon entropy. So this damping factor makes the low probability differences, fold changes, also picked up by the distance. And the idea, again, of using metric MDS instead of eigendecomposition is so that we only get 2, 3, or however many dimensions you want. And it's doing the best job it can to preserve all this information in low dimensions. So that gives you PHATE. And here again, these are ground truth examples where we generated the stuff that's on the first column. So we generated this, and we see, what PHATE is giving us versus what other things are giving us. So sometimes the other methods do pretty well on sort of like the Gaussian mixture model. Sometimes they again do weirdly, like tSNE is putting the green and red in between the blue and orangish color. Sometimes they're very noisy. Like isomap can be very noisy because it's doing geodesic distances, shortest path distances. So shortest path distances will take every shortcut they can through every outlier they can. You can see things like force-directed layout that are just trying to like splay things out, and you get what one of my students called sneeze spots from it, where it looks like the points are sneezed out. There's other methods called monocle that fit trees to data, and it's not quite appropriate, but some reviewers asked us to compare against that. And then you can imagine that it does the same thing to biological data. So there's different types of data we visualize here. The first is our embryonic stem cell differentiation data. The second is retinal bipolar data, which they mainly thought had a lot of clusters, but we can see that the clusters have some progression within them. And then there's hematopoietic stem cell differentiation data in A. There's iPSC reprogramming in E. And when they're grayed out, it means those methods don't scale, so we had to subsample them to begin with, which turns out to be a huge problem for most of these. Whereas this kind of diffusion process can be made really fast using some tricks. This is a visualization of what PHATE was trying to do. It's trying to keep global structure like PCA, but it's also clarifying local structure because it can denoise in nonlinear directions and find nonlinear directions of dimensionality. So you get more fine-grained detail than you would. And you can sort of zoom in on those fine details, and I don't have a zoom-in slide, but we can show, for example, that this orange branch, that the way it's splitting is actually real, and there's some biological conclusions about what the differences between those two retinal cell types are. So what can we do after we run PHATE? You can do a lot of different things. You can cluster on the PHATE dimensions. We compute intrinsic dimensionality to find sort of branching points. If you have a lot of 1D trajectories in your data, branching points have higher dimension locally. We can use these branching points in conjunction with the clusters to come up with sort of lineages or regions of analysis. And so we gave this kind of analysis to our collaborator at the Yale Stem Cell Center, and she was able to play with this tool and identify a lot of different lineages. So she came up with this tree of lineages, and there are several intermediates here that she did not know. So this is truly a tool for people to visualize data, their own data from their own domain, and figure out what's in the data and use it for further analysis. So we were able to sort out some of the populations that she was interested in, and then she can fact sort them and figure out what to do with it next. So the final project that I'll talk about, it kind of ties all of these things together. Yeah.
So it seems like there are two big things. One is sort of like using the diffusion to do the smoothing. Mm-hmm. And the other one is you replace the...
To do sort of, yeah, go ahead.
And then the other one is you use this informational divergence rather than Euclidean.
Yeah.
System. So
Exactly, that's exactly.
So I'm wondering whether you guys tried to ablate the two aspects and see which one.
So actually all these steps are essential. There is a random collection. Each of these steps is essential for getting the end result. So if you remove the diffusion, it doesn't work. If you remove the informational distance, it doesn't work. So all of these are essential steps. Yeah.
And if you don't go from, we validated that every step is basically necessary. Do we still have that figure in the Markov paper? No, there's always space constraints when you submit it to the user.
I'm just wondering, because people use tSNE for like gene expression clustering, and there I can see the one big difference is that the cell count, the transcript count is not as noisy as this thing, right? So
no, it is. It can be. So I think tSNE's maybe okay for some kind of clustering because it kind of keeps local neighbors together. But if your data doesn't have that kind of cluster structure, and it keeps them together at a very particular granularity. The granularity is determined by the perplexity you set in tSNE. So basically, at that level of granularity, it's kind of splitting your data out into clusters, and I think that's an okay use case for tSNE. But it's not an okay use case if you're trying to find the relationships between your populations, which clusters are actually close to each other. Because also the KL divergence penalty is very lopsided. It cares if you put close neighbors far, but there's no effective penalty for putting far things close or in any configuration. But what tSNE does that PCA doesn't do, for example, is it does squeeze variability into 2 dimensions, but it does it with stochastic gradient descent. And so that's a little random. So if you run tSNE many times, you'll get different results. So the next part of this project I'll talk about is, so I've talked a lot about how really understanding the shape of the manifold helps you visualize, helps you figure out the differences between experimental conditions, it helps you restore transcripts. So can we do some of this with deep learning, which adds to it all the other features that deep learning has, such as even increased scalability and the ability to do many things at the same time because you have some sort of deep model through which you can process your data.
By the way, PHATE is very scalable.
PHATE and Magic are both very scalable. So, and David worked a lot on making them super scalable. So I'm sure he wants me to mention that. But I'm sure you guys know that in deep learning you can farm it out to a bunch of GPUs or something. The more GPUs you have, the easier it is to parallelize this. But if you have a lot of CPUs, it's fine. So SAUCIE is an autoencoder-based framework, and that's what the A stands for. So we think really hard about making these acronyms. The idea of SAUCIE is actually to find these kind of patterns emergently in a neural network instead of using some kind of diffusion-based algorithms based on constraints and regularizations. And what we're trying to do with SAUCIE is we're trying to go to the realm where there's massive amounts of data. Jonathan was saying today that there's massive amounts of TCR data, for example. So if we have massive amounts of data, like single-cell data collected on many, many samples, we want to be able to run it through this system and come out and read a lot of patterns off the fact that an autoencoder neural network is essentially a nonlinear dimensionality reduction, as long as you have nonlinear activations. So again, some of the things that we designed SAUCIE to do are, from the ways the different layers are regularized, we can read these patterns off the data. So actually, people always complain that you don't know what the inside of a neural network is doing, and that's not true in our case of our neural network, because on the outside, it's doing one interesting thing, which is denoising data, but other than that, it's just recreating its own input. That's what an autoencoder does. The internal layers are good for visualization. We can read clusters off one of the layers. We can batch align or batch normalize them. And then we can also recreate the output. So those are the different layers. And then finally, it gives you sort of the nonlinear dimensionality reduction on particular patients can give you stratifications of the patients themselves. So I guess you don't need an explanation of deep neural networks. An autoencoder was first introduced many years ago. I don't know when it was first introduced. It was popularized in this science paper in 2006. And the idea is very simple, that the only penalty that the neural network natively enforces is that the input be recreated at the output. And so this makes it unsupervised. You don't need any labels. And it means that if you have reduced dimensions in the middle, that they have some kind of dimensionality reduction going on. So this is more specifically what SAUCIE looks like. So SAUCIE has an MMD penalty, or a penalty to align batches, enforced on its lowest dimensional layer. So the idea here is that you treat the batches as different distributions and align them. There's a layer that's two-dimensional that you can use for visualization. There's a layer that has a special regularization that we call the information dimension regularization that binarizes activations or makes them easily binarizable. And there you can read clusters off the data. And finally, the output layer denoises the data because it's trying to recreate the data after throwing off all these dimensions in the middle.
Could you talk about what your reconstruction loss is? Because I mean, based on the stuff that you showed earlier, it seems like the particular error metrics that you use make a lot of difference in terms of how your algorithms perform. So I'm curious, are you using simply Euclidean loss, or is this trying to use some of the probabilistic type of losses?
So SAUCIE is not stochastic, although we have other stochastic neural networks. It is using a simple reconstruction loss, like mean squared error.
Euclidean loss.
Yeah, but there's penalty terms due to the regularizations. So there's
And the other layers.
yeah, but they're all added to the, yeah.
So for example, that middle layer on the right, like commonly with autoencoders, basically just reverse the encoding network to do the decoding network. But it looks like you're actually doing something different.
We're doing something
constraining
different.
the output
Yeah.
network.
Yeah, and I can go over that. I think I have some slides to go over how we constrain it. So we constrain it so that layer is giving us.
Don't really use tight weights anymore.
Some people do probably, but
Yeah, but it
No, but I mean, even if
seems
you
to
don't
not work as
decide
well
ways,
as the
people often just sort of use the same network architecture, just flip it.
Right,
Which
that's
is
true.
true,
Yeah,
yes.
yeah. But
But you can add new constraints and
Yeah.
regularize yourself.
and actually I don't think that layer size is similar as is shown here. I think it's a fan-out layer. It's sparse. So the visualization, again, you can compare to these other methods like PCA, tSNE, and things like that. You just take the two-dimensional layer and visualize it. This is the representation for the clustering. So the idea is that if you have some cluster of cells, you want to be able to read off which cluster they're in. And the within cluster variation is only stored in a small range on top of this binarization. It's still there because it recreates it, but the majority of the signal that these nodes are giving is which cluster it's in. So this particular cluster is encoded by basically 0, 1, 1, and a different cluster could be encoded by something different. So the way you do this is we penalize for activation entropy as in von Neumann entropy. So if you take all of your activation values and you treat them as a probability distribution and you compute Shannon entropy on it, if all your nodes are activated to roughly the same level, then you'll have high von Neumann entropy, if you can call it that. If one of them is 1 and a lot of them are zeros, or a few of them are 1s and a lot of them are zeros, and you have low activation entropy. And so this gives you sort of binarizable entries or activations. So with no regularization, you see a lot of things are activated middle, high. L1 regularization, which a lot of people use, just avoids the very high value, and then they're pushed over, and you still have a lot of entropy there. But our regularization gives you a binarizable shape. So if you round it or actually binarize it, you get cluster level information rather than cell level information from this. And that's the main idea of the regularization on the layer that reads off the clusters. For the batch effects, this is an embedding of patient 1 versus patient 2, and this is a dengue patient data set. And we use a probabilistic distance that's been used a little bit in neural networks, but not too much. It's called maximal mean discrepancy. And you can compute this at the batch level. You're computing that batch 1 have minimal probabilistic distance to batch 2. But it still has to reconstruct and do these other things. So it encourages it to sort of minimally align the data and not totally re-scramble it. So for the red and the blue are different patients. So the embedding is mainly organized by patient, which we don't want. We want to see, we want the batch effects between when the patients were measured, what time of the day, all of that to go away. So after MMD, you see that it's organized by cell type. So now we can see how much CD3 positive cells both these patients have and things like that. I don't know. So we tried this on this dengue data. It was from NIMHANS in Bangalore. It was the neurovirology department at NIMHANS. So they gave us, it's something like 60 patients, and they visited 2 times, and then there's an additional number of patients who came with them, and they're the healthy controls who are living in the same environment. And so there were 180 files. Each of them have many, many cells, and so totally this data was 20 million cells. And you had to batch normalize between 180 different files. And there's the few batch normalization tools that are out there don't really normalize at this scale. They do 2 conditions or something like that. You see what the cells look like if you combine all the acutes and the healthy versus the convalescent. So convalescent are the acute people. They came back some weeks later, and their cells are starting to look a lot more like the healthy. It's just a sanity check. But you see that there's no sort of batch effect. You can't see each of the acute patients separately or anything like that. It's a unified manifold. So on this unified manifold, you can go ahead and cluster like it's one data set, which wasn't possible before. There's other methods that will cluster totally separately and then try to match these clusters, and it turns out to a huge problem. Instead, you can just cluster this data as if it's one data set, and you come up with these populations of cells. We find that some of the populations of cells, for example, are very interesting. There's the TCR gamma delta population, and we saw that a lot in the acutely infected patients, that they have sort of like NK cells almost. They have some innate activity. They show signs of innate activity. And this is a rare population, but we can still pick up a systematic difference. So then you can see these signatures of these different cell types for the different patients that are acute, convalescent, or healthy. And then you could embed it again with an MMD-based distance. You see that all the patients themselves are stratified, where the left side of the embedding is a lot of the acute patients and the right side is convalescent, and you see people are convalescing to different extents and things like that. And then you can associate this with the proportions of different clusters that you see in your clustering. And of course, the point that I was making is that you're running all of this through these neural networks that are tricked out or whatever. So it should be very scalable, and we actually do find that it's very scalable. So we've compared against, so k-means and phenograph are clustering methods. tSNE sort of a clustering, but also visualization method. PHATE, this is the old PHATE, sorry, compared against the non-super fast PHATE. PCA, neural network completion, MNN is a type of batch normalization, diffusion maps, all of these things, and it's super, super fast, faster than everything besides, I think it was PCA. And that's because now PCA is done with random projection matrices really quickly. But PCA doesn't give you very, very good results. If you're randomly, linearly projecting, you're losing a lot of information. So quality-wise, SAUCIE is way better than any kind of PCA because it does so many other things, and it's a nonlinear method. Finally, it's just because I added one slide just because Jonathan was talking a lot about dynamics. So we have a neural network that we're training also to predict dynamics in these things. And this network is generative and it's stochastic. So we're calling this the transcoder. And because PHATE and these other methods allow us to pick up trajectories, we're hoping to mimic these trajectories and find some deep representation of the logic that's used to make these transitions. But that's an ongoing project. So we're hoping to test this on some kind of evolution systems, protein folding systems, and things like that. Those are the main projects that I want to talk about. The main idea is we pick up a lot of structure that could otherwise go missing in data by using very careful nonlinear dimensionality reduction and manifold learning methods, using graph signal processing and also deep learning. And we believe that these have wide applicability, much beyond what I said to the specific data sets that I was showing it on. I also have a lot of talented students and postdocs in my lab. So I'm going to try to get them to intern at IBM. Let me know. They were doing a hackathon then, and so I just took pictures of them at a hackathon. And actually, I think they were having a lot of fun, even though they look funny. And of course, we collaborate with a lot of people at Yale, including immunologists, infectious disease people, stem cell biologists, neurologists, Yale Center for Genome Analysis. All the software I've talked about is on GitHub, maybe besides transcoder. Papers are on bioRxiv. Some of the papers on related topics are in NIPS and ICML. A lot of them are on bioRxiv also, or regular archive. You can feel free to email me.
These for PHATE and Magic in R, MATLAB, and Python.
Yeah.
SAUCIE is just in Python. It uses it for.
Yeah, we noticed that we have to, even though nobody in my lab likes or uses R, biologists won't use it unless you code it in R. We make someone do it. So let me know. Are you an R fan? No. Okay. What do you guys code in? Your own languages?
Python, sure.
Python, yeah. Start an argument.
What century is this?
What century is this? Yeah. If you have any questions, let me know. We have sort of additional slides on these types of things. Yeah, go ahead.
Yeah, so in terms of scalability gain, I think of it largely like...
I'm sorry, what?
So you show the scalability gain, right? So can I think of it as like in Magic and PHATE, you need to think about this pairwise distance, and that's where you get the blow up, whereas in neural
that's effectively where you
we've
get
solved that.
Yeah,
Some
you can answer that.
Yeah.
Okay. But why in that case?
I do have a slide that kind of shows that.
The supplemental slide deck.
You have, well, yeah, sure.
But also a related question is that, can you imagine using a neural network embedding way to do the same thing like when you do the diffusion? Like
we do use the neural network to do the same thing. I'll show you.
Like right now you use a diffusion to...
So we don't, the neural network like SAUCIE doesn't explicitly...
So the last layer of SAUCIE actually gives you the same kind of reconstructions as Magic. And it most closely compares to Magic, more so than to the other imputation methods. And this is in the SAUCIE manuscript. But this actually shows that this neural network also learns the manifold without diffusion and without us having to define a distance metric.
But it's some other type of low-pass filter.
Yeah.
You can see it as. Just not implemented with diffusion on a graph, but using cascades of
in NLP we have
nonlinear
very similar thing where you have a knowledge graph. Like your knowledge graph could be a relation between the entity, and then traditional people use random walk to compute a diffusion probability.
You mean you're talking about like word vector or something like that?
Yeah. Nowadays we will embed it into embedding, and then you can compute any arbitrary thing without doing any random walk. Because a random walk would be expensive.
Right. Yeah. Even though we have solved that.
Oh.
Yeah. So here you can talk about that. You want to talk about this slide, right? Yeah.
Oh yeah, nice.
You can explain it.
So instead of computing all paths between points, we're constraining the paths to go all through specific landmarks. We fix a number of landmarks, but we pick enough of them to accurately sort of describe the graph. So now we essentially get a linear complexity.
So now the diffusion goes from original points to landmark points to original points, but if you do the matrix association correctly.
And the landmarks are chosen to be sort of uniformly representing the whole graph. And so we lose very little information but we still can exclude the vast majority of paths.
So you literally random sample the landmark?
No.
Or
we actually use a spectral clustering approach to pick
okay.
the
So
landmarks.
you use one approximation
using
first.
fast random SVD, so it's all super fast.
I see.
So it's
essentially
the whole thing is linear complex.
So it's almost like a little bit like a random projection.
Yeah, we do random projections to pick the landmarks, and then we compute the paths to go through the landmarks.
Interesting. That's very interesting.
Yeah. And it works almost exactly get the same result as the original sort of complete version. So yeah.
Guys, you can look up PHATE either on bioRxiv or on Cell Sneak Peek, and it'll show these comparisons and thousands of supplemental figures and stuff like that.
So the spike in the runtime there, is that when you're not using as many landmarks as there are points?
Yes, exactly. That's a very good observation. So we use, I think by default, something like 2,000 landmarks. So when you have fewer points than that number, there's no point in doing that. And yeah, so only really in large numbers does it sort of help to pick the landmarks. That's exactly why you have that problem. Yeah.
The other motivation for SAUCIE is not just scalability, it's also that all these tasks are implemented on some kind of unified representation of the data. So we find that some clustering methods, they might represent the data as a graph or some kind of cloud, and then you have a batch normalization method that represents data as canonical correlation dimensions or something, and there's very disparate stuff, and the different features you're reading off the data are not made from the same representation, so they may not correlate with each other.
And tSNE in your data, there's no guarantee that tSNE picks up the same clusters as k-means because they have just different algorithms, different representations. So that was one of the reasons.
Yeah, so when you do stochastic gradient descent, you pass the gradient all the way back, so all the layers are sort of coordinated.
And here we have one representation that kind of guarantees that you get the same clusters as your visualization.
Mm-hmm. So that clustering, where you use the information, the entropy measure to constrain, is that, so that's going to effectively pick, push you towards picking a single cluster at that layer, right? So you've got N.
You're forcing the nodes to become binary, sort of digital, either zeros or ones.
But even more so, wouldn't it tend to force them to have a single one?
Not
a
a
single one would be like L1, so sparsity. Sparsity would just try to make as many nodes go to zero. We're not doing that. We want the nodes to be zeros or ones, so we want it to be binary and digital.
So the entropy is being computed on each node independently?
So each node becomes a probability.
Oh, okay.
And then we do Shannon entropy. It's actually not von Neumann entropy, it's just Shannon entropy.
I'm calling it von Neumann entropy because it's like an entropy of values rather than probabilities. That's why I was calling it that.
So yes, it's within each node or across all the nodes?
It's across the nodes.
Across the nodes, yeah.
Yeah, but we don't see that it gives us a single one because it's not strictly a sparsity penalty. So usually the neural network will not make it one hot. It'll make it low entropy given that it can still reconstruct. So usually the vectors are, 3, 4 ones.
Yeah, because there's just one you could put in a softmax where it would kind of end up pushing you to pick one.
Yeah, like one hot encoding. You could do that.
But we don't usually do that.
We find
push
that
it to one hot.
When you do one hot, you have to pick the number of nodes, which will give you the number of clusters. And in this case, actually, we don't.
No, we don't know the number of clusters.
No, that's what I'm saying. Here, we don't know the number of clusters, so we don't have to define. We have to set the number of clusters that we get out of it.
But we can overall tweak the granularity by upping this penalty or downing this penalty, but we don't know exactly how many. Like we're not saying like 10 k-means give us 10 clusters or something like that.
So how do you set this coefficient for the regularization right now?
So that's sort of a variable. It depends on if you want really big clusters like T cells versus B cells versus monocytes, or if you want to go inside the T cells and see the different subtypes.
Also, it's pretty robust. Like we see that when you change it, the biology is pretty same, or whatever test case we have, like Gaussian mixtures or whatever. So it's pretty robust against those parameters. Yeah.
Cool. Well, thank you guys.
Thank you.